Welcome to this lecture on the topic of agronomy and sampling. This subject is part of the agricultural degree offered at North Melbourne Institute of TAFE, which is based in Australia. Please visit our website for further details on the subject, courses and education products we offer. www.nmit.edu.au. My name is Dr. Nikki Cooley. In this lecture we will be spending some time on sampling. We will be looking at soil sampling and plant sampling. We will look at spatial variation and temporal variation and the role that we need to consider with respect to sampling. We shall also be considering precision sampling. Sampling is used in agriculture, agronomy and extensively in precision agronomy. It is essential that you understand the requirements for sampling, as this will often frame the observations and their meaning. Incorrect sampling can lead to significant costs, resulting in costs from time, cost of misuse of resources, and cost of incorrect management. Before sampling, consider the protocol and that it meets your requirements. Consider the sampling replication and ensure that it is adequate. Consider the location of the sampling, either across a plant or across a paddock. Wendell Berry, in The Unsettling of America, Culture and Agriculture, was quoted as saying, "The soil is a great connector of lives, the source and the destination of all. It is the healthier and the restorer, and the rescuer, by which disease passes into health, age into youth, and death into life. Without proper care for it we have no community, because without proper care for it we have no life." This quote sums up the importance of soil, and indeed it is very true for agronomy as well. We would sample the soil because we would be interested in the nutrients and possibly the microbes in the soil which allow some nutrient uptake, the pH structure and texture, all of which can affect our plant growth and development, the water holding capacity and soil water content. This is an important component of growing crops in Australia, and salt and salinity. Characteristics that are linked to plant health and other crop productivity aspects. The ideal time to soil sample is in the autumn or summer, after crop removal and before planting. It is advisable to use a soil auger. Take large samples, hard grounds and deep samples. Commercial examples of soil sampling equipment is found on the website below, www.soilsample.com. We will not go into a lot of detail about these techniques as they are covered in your soil and nutrition topics, which you will study later on. How to conduct soil sampling. Firstly, you should select an area to be sampled. Consider the sample location options. If possible, record the GPS data, that is the location, the time and the date of the sample. If a crop is present, record the growth stage of the crop, record the weather, remove excess plant residues. When using a probe, push the tube into the soil to the desired sample depth and collect the individual sample. The number of compostuates will depend on the soil data requirements. Depending on your required replication, you may test individual cores or you may bulk the samples. Ensure you make a note if you need to sample across soil horizons. If horizon data is required, identify the horizon and cut this area and mix well in a bucket. Where soil core bulking has occurred, select a composite sample from the mixture for testing. Place the composite sample in a sample labelled bag. Allow the sample to air dry to stabilize nitrogen. The sample is then ready for analysis. In conclusion, proper sampling is a foundation for a proper nutrient plan. soil nutrients availability can be determined, and this information can be used to compile a plan for your nutrient application, both timing and concentrations. Correct understanding and application of soil nutrients should improve crop use and reduce waste. pH information in some crops is essential, and the cation exchange capacity can also be a useful piece of information to have. Select your sampling method and frequency to reflect your farm and crop requirements. Standard procedures ensure optimal interpretation of the data. If possible, employ precision sampling practices and keep good records. Understand that soil variation does exist. On Moodle, you will be supplied with a soil sampling and laboratory selection guide. Please read the following resource, which is produced by Montana State University. Please write a paragraph in your notes about this protocol. Is it a good protocol? If so, why? Is it easy to understand? Then write a second paragraph on how you might apply this sampling protocol at the Yangying Teaching and Research Demonstrator. Does the protocol need modification, and if so, how? Do you need to consider your replication? Please bring these notes to your next class at Yangying. There are a number of different methods that you can employ to sample over your field and your paddocks. Simple random, systematic, and stratified random. A diagram of each of these protocols can be shown on your screen. There are advantages and disadvantages for using each of these sample location methods. Precision sampling is methodology that has developed in recent years, particularly with the innovations and uptake of GPS. It enables sampling methods that employ site location. It aids sample history, the location of the sample, the time and the date of the sample collection. It enables collection of data that can result in precision fertilizer application and yield monitoring. This improves fertilizer efficiency. Data generated can be used to generate spatial maps. This sits under the discipline of precision agronomy. Now we will look at crop sampling. There are many reasons why you may want to sample your crop. You may want to determine your nutrient content, or you may wish to predict your yield. Crop health is also another reason why you may take some samples. Crop sampling is used extensively in precision agronomy. It allows optimal inputs and sustainable agriculture. It has benefits for the environment as you have a reduction in chemical and nutrient wash-off. And knowledge is gained by the farmer as you improve your understanding of your ecosystem and your variation. There are some sampling considerations that are the same in soil as they are in crops. This is things like replication and the locations of the samples in the paddock. But in crop sampling, you also have to consider such aspects as plant tissue age, usually juvenile versus fully expanded leaves. You need to think about the plant tissue location. Is it a leaf? Is it a stem? Plant tissue handling and how you process the tissue once you have removed it from the plant is very important. Do you require it dried or fresh? What is the correct storage before processing? And is time before analysis critical, as it can be in many crop sampling analyses? Temporal variation is really just variation that changes with time. The definition is a variant that is a function of time. It is significant in both crop and soil sampling. There are several causes for temporal variation, and some are obvious. Growth in the crop plants is continuous, so you have different changes that are occurring depending on the growth stage. In the field environment, resources and inputs are changing constantly too. Some of these changes are instantaneous, while others cause accumulative effects. Other aspects that may not be as obvious is that crops will contain circadian clocks and circadian variation across the crop. Please read Plant Circadian Rhythms, which was published in the Plant Cell Volume 18 in 2006. After you have read this article, please ensure your notes include a definition of what a plant circadian rhythm is, a description of Figure 1 from the paper, which is shown below, a description of ZT and some examples of plant circadian rhythms, and at least one example of a circadian gene. Place these notes on circadian clocks and photoperiodism with these lecture notes to complete the full content of this topic. The figure on the slide illustrates the change in fresh wheat weight and age of a wheat seedling. You will observe that as the age of the seedling increases between 0 and 4 days, so does the fresh weight increase. Not only fresh weights of plants increase over time, but so does dry weights. The illustration on the slide demonstrates the increase of dry weight accumulation in wheat leaves from germinations to 115 days after sowing. This demonstrates the relationship with nitrogen application in irrigated wheat. You will also see that the initial phase of growth is linear, and that growth slows down, dry matter accumulation per day, as the phenological stages increase, or as the days after sowing increase. This figure is a very good example of showing how wheat growth increases and how this is linked to nitrogen uptake. The chart shows that nitrogen uptake changes over time and that it is quicker or more uptake in certain phenological stages than others. Later growth results in more nitrogen uptake. The quantitative relationship of wheat nitrogen uptake over time and different phenological stages is demonstrated in the figure on the slide. You can see that at jointing, which occurs in March in this crop, there was a massive uptake of nitrogen. There are several aspects that you have to consider in sampling when you are looking at temporal variation. You need to think about the sample frequency. This is specific to the sample requirement. For example, you could collect soil moisture 15 minutes across a day or once a day. Depending on how often you collect it will depend on the interpretation that you can make from this data. Soil nutrients are specific to the season and the plant development stages, the change of crop, and sometimes linked with the results of soil moisture. Plant nutrients, again, are to be measured at set development stages. There is not much point in measuring them at the beginning of seed germination, just after seed germination, especially if you've only just applied nitrogen at this point. And if you were measuring for yield several weeks before harvest, the accuracy increases as the harvest date becomes closer. Spatial variation is another important component to consider when sampling. Spatial variation exists in all living systems. Let us start with the soil. This is subject to spatial variation, which is caused by many factors, and these can include differences in the structure of the soil across a paddock, differences in the texture, different microbial communities across a paddock, variation in the nutrients, both the concentration and the quantity and quality, the pH and the rainfall. With variations in soil, crop growth and development variations can result. Climatic variations can occur in the paddock, and this can depend on the topography of the surrounding area, but also the topography of the plants. Often you will see microclimate conditions. These can result in variation to crop growth and development, and perhaps the most obvious are those of the edge effects. Rain is perhaps the most predominant significant climate variation found. Variation to other important inputs, applied nutrients and management practice can all increase spatial variation. The amount or degree of spatial variation can be different between different crops, soil types, climates and management practices. Therefore, it is important to try and understand your spatial variation and how that may impact on your agronomy. Understanding your spatial variation and adjusting your management accordingly can reduce your inputs, allow more sustainable farming practice, and improve your economic return if done correctly. These observations on spatial variation and its importance have been quoted in many papers. Such one paper is an abstract from Thornton 2009. This will be put on Moodle so that you can read it more clearly. Please take the time to read this abstract and take away the main points it's trying to make. A second paper, written by Charles Edwards in 1987, discusses the analysis of spatial variation in specific nitrogen content of leaves and what this means. Again, please take the time to stop this lecture here and read this abstract. Please make note what the scientific contribution of this statement is and what lessons you can learn from this research. Let us discuss some of the considerations of spatial variation and sampling. In order to assess spatial variation, you need a number of sampling locations. The exact number will depend on what you're sampling for. There are a number of methods that can be used. You can have a set number for each paddock, which is randomly or assigned in a grid. Or you can have a set percentage, that is, you're trying to represent a certain percentage area of the paddock. And again, you can select your sampling points either randomly or by a grid. The image on the slide is from Boll-Will Turning Precision Technology into Agronomic Solutions by Roundup. It is a georeference soil sampling via a grid method, and it gives the ability to paint an accurate picture of the information that precision applications are based off. The sampling distance in this diagram is between 0.4 and 2.2 hectares. I have included a commercial example of software that helps you with sample strategies. This is AgroStudio Map, and it is a field boundary and soil sampling application designed to integrate directly with MapShoot's AgroStudio Precision Agronomy software. Please note this is an example of the technology available and not necessarily an endorsement of this product. One of the exercises we are going to complete in this subject is to learn how to sample for modelling. This is important in yield prediction. Please read the following cereal growth stages. This guide is a highly respected guide to cereal growth stage and assessment. It is important that you are able to assess the growth stage of these cereals. How would you be able to apply this protocol to the Yang Ying Teaching and Research Demonstrator experiment? Think about the wheat plants that you have seen and how you will be required to understand the different stages of their growth. Please ensure that you take time to review this manual before you attend the next visit to the Yang Ying Teaching and Research Demonstrator. The second piece of literature on sampling for models I'd like you to read, please, is the Overcoming Production Constraints to Sorghum in Rain-Fed Environments in India and Australia. This paper outlines extensively the methodology to be used for sampling for models. This will be a resource we will use continuously while we are at the Yang Ying Teaching and Research Demonstrator. Please familiarise yourself with this information before your next visit. We will be discussing this in the teaching and research demonstrator context. Other aspects that you might consider when selecting a sampling strategy is the topography, the paddock management history. For example, do you know what the nutrient application has been over years, and can this influence your sampling? The fixed interval or grid sampling, and there is also a stratified random sampling that can be applied to look at these different inputs. Please take some time to think about how you might sample both the plants and the soil in the Yang Ying teaching and research demonstrator experiment that you helped set up in the first week in this subject. Would you use a random sampling technique, or would you use a fixed interval or grid sampling technique? It is worth noting that the experimental area is quite small compared to a paddock. Therefore, should the reduced size be reflected in how you sample? We will be having discussions of this in the second week of the practical visit at Yang Ying Teaching and Research Demonstrator. The notes here will complement your visit to the Yang Ying Teaching and Research Demonstrator. In this summary, we have begun to understand what sampling is, the differences between soil and plant sampling, how sampling should be adjusted if we are to take in spatial variation, and how sampling should be adjusted if temporal variation is our aim. Increasing your sampling frequency and understanding the optimal sampling protocol are very important in both the interpretation of your data and the ability to use this information effectively in management decisions. This brings us to the end of this lecture.